stretch. And that final stretch is going to culminate in a final on Thursday, December 9th at 6 p.m. And for that final, you will be allowed to use a note card that I provide. Okay? So let me just say a few words about the note card and also a little bit about what we're going to be doing this week. So I do anticipate we will finish all the material. We were a little bit ahead when we had class canceled. I've also posted a lecture of last year that will cover the gluconeogenesis. I'm going to go through that fairly quickly today. And obviously, if you have any questions with respect to that, let me know. Now, the rules on the note card are as follows. You have to get the note card from me. Okay? And yes, I have a way of telling. So you have to get the note card from me, that's number one. And number two, even if you don't want a note card, you have to have a note card on the day of the exam. Okay? So everybody has to have a note card. If you don't have a note card on the day of the exam, you will lose points. All right? You will have to turn the card in to me when you're done. All right? So if you're really attached to it, make a copy of it or something like that. My reason for doing this is that this way you don't hand it off to your roommate, and next year your roommate has a card that they're using but that they didn't fill out. And I believe that filling out the note card, you're going to learn a lot of stuff. And I want the roommate to have that advantage of filling the card out themselves as well. Okay? Questions? What if in all the chaos of dead week you lose your card? Then you come see me and say, Kevin, I was stupid and I lost my card. And then we'll deal with that. How about that? So my advice is don't lose the card. All right. So the cards are here. One card per person. If you want to come up and get them, let's do that. Spend a couple minutes. And everybody has to come get their own. So one card per person, okay? One, absolutely. One card per person. What'd I say?
Oh, okay. I get it for...
No, that's why I said one card per person. You can write on the side if you want to. I've seen people that write small enough they can almost do that.
So it's two-sided?
Yeah.
Are you going to show the grade distribution?
It's on the web. What's that?
Are they watermarked?
I can't tell my secret. What's that?
I said it's a radioactive tag, right?
Can't tell my secret. How you doing? Good. What? Oh, okay. What did you do? You got to quit kicking people. Okay.
Had a good lunch today?
Yeah.
All the extra.
All the extra turkey. You can use both sides. You can use the edge if you want. Okay, folks. So. This is the only time these will be available in the class. So if you miss class today, then you will need to come see me about getting a note card. And that way I get an interesting measure on who's coming to class, don't I? Okay. Now, as I said, I'm going to go through gluconeogenesis fairly rapidly today because I have posted a previous lecture on that. And if you find that things are a little fast or confusing, then please do come see me, and I'll be happy to give you a little bit more time separately on that. So, as I said, the exam is in here at 6 p.m. on December the 9th. That is Thursday of finals week. And as I said, you will be required to bring a note card that you get from me. So even if you don't plan to use one, you need to have one there. So that's important for everybody to understand. You will lose points if you do not have a note card there that you got from me. And obviously you need to get it from me. All right. So that's pretty much what I want to say. The exams are obviously graded. I'm sure most of you by now have gotten your scores. The scores and the grades are posted online. I'm not going to go through that to save a little bit of time. And again, as always, if you have questions with that, let me know. I did see that the TAs were very harsh on one question, number 14, I believe, in section 1. And so the answer key says GTPAs, and if you didn't write GTPAs, they didn't give you credit. So if you said something like breaks GTP to GDP, then probably you should have gotten some credit. So if that happens to you, let me know. I'll be happy to make adjustment as appropriate. Whoa, a lot of people. That is a correct answer. So, I mean, I don't know why I need to talk to them. I didn't know until a couple students pointed out to me that they had that answer. But you do have to say GTP broken down to GDP. That if you say that, you should have gotten credit. Okay. So last time I got started talking about gluconeogenesis, and that's where I'm going to pick up here very briefly, going through that. As I mentioned last time, gluconeogenesis is very similar to the reversal of glycolysis. There are 7 reactions in glycolysis that are identical but reversed in gluconeogenesis. And not surprisingly, those 7 reactions don't have the big delta G changes that we see in glycolysis. So it makes it fairly easy for the cell to reverse those. Now, one exception, of course, is the aldolase reaction, which in the gluconeogenesis direction has a delta G 0' of plus 20, but the overall delta G for that reaction is very close to 0. So it makes it very easy for the cell to switch the direction of that reaction as conditions merit. Now, there are 3 enzymes in glycolysis that are replaced by 4 enzymes in gluconeogenesis. And those, I started down here last time talking about pyruvate kinase. So pyruvate kinase was the enzyme that catalyzed what I described in glycolysis as the Big Bang reaction. And that Big Bang reaction is more than the cell can simply reverse. So what the cell does is it substitutes 2 different reactions for the pyruvate kinase reaction. The first reaction catalyzed by an enzyme known as pyruvate carboxylase. And pyruvate carboxylase converts pyruvate into oxaloacetate. That requires addition of a carboxyl group. And the addition of the carboxyl group is made possible by a coenzyme that pyruvate carboxylase uses called biotin. Biotin is a coenzyme that's commonly involved in carboxylation reactions. That reaction, catalyzed by pyruvate carboxylase, requires ATP. And since we start, obviously, with two pyruvates to make one glucose, then we have two ATPs along the way to get back up to glucose. In the second reaction that's different, oxaloacetate is converted to PEP. This reaction requires an enzyme called phosphoenol pyruvate carboxykinase, or as you'll likely call it, PEPCK, or PEPCK. That enzyme converts oxaloacetate into phosphoenol pyruvate, PEP, and requires GTP. GTP has essentially the same energy as ATP, so for all intents and purposes, we're looking at this point having gotten to PEP using 4 triphosphates to go from 2 pyruvates to 2 phosphoenol pyruvates. All right? So we see, well, and what we will see is that gluconeogenesis requires more energy to make glucose than what the cell gets out when it breaks down glucose, and that's not totally surprising because these reactions are not 100% efficient. Well, once we get to phosphoenol pyruvate, we see a reversal of steps. PEP goes backwards to 2-phosphoglycerate. 2-phosphoglycerate goes backwards to 3-phosphoglycerate, etc., climbing the ladder, as it were, until we get up to fructose 1,6-bisphosphate. And it's at that point that the cell uses another enzyme to bypass that energy barrier of glycolysis. Now, at first glance, it might not seem like it's really doing a different reaction than the reversal of glycolysis. For example, simply changing the enzyme, but having the same reaction would still have the same energy change. So not only does the cell have to change the enzyme, but the cell must also have a different reaction. Well, the reaction that is involved in glycolysis requires ATP. That is, ATP is necessary to put a phosphate onto fructose 6-phosphate to make fructose 1,6-bisphosphate. Reversing the reaction simply involves removing that phosphate. So instead of regenerating ATP, the cell is using the energy of that fructose 1,6-bisphosphate to drive the reaction. The enzyme that catalyzes that is called fructose 1,6-bisphosphatase, or as you can call it, F16BPase. So a phosphatase, you remember, is an enzyme that removes phosphates from things. It does not put it onto anything else. So in this case, it's removing the phosphate from fructose 1,6-bisphosphate and just leaving phosphate floating around in the cell, which is a very common phenomenon. This also makes sense in for another reason. Think about this. When the cells are making glucose, what is their ATP state, high or low? It's going to be high because when they have a lot of energy, they want to store that energy as glucose. So ATP levels are going to be high. By not resynthesizing ATP, the cell is not adding to that already high level of ATP that's present in cells. Okay? The cells are already saying, I want to reduce ATP levels, so there's no reason to reverse the reaction of glycolysis and try to remake ATP, because that would only add more ATP. So the cell avoids doing that and simply clips the phosphate off and makes fructose 6-phosphate. Okay, well, the next reaction, of course, is a reversal of the glycolysis reaction, and that produces glucose 6-phosphate. And that leads us to the final enzyme that is different in gluconeogenesis compared to glycolysis, and that is the reaction that's catalyzed by the enzyme glucose 6-phosphatase, or as you will probably refer to it as G6Pase. Now, the first enzyme of the pathway was pyruvate carboxylase that I said was found only in the mitochondrion. The last reaction of gluconeogenesis is found only in the endoplasmic reticulum. And so all the other reactions in between there all occur in the cytoplasm. So the last reaction occurs in the endoplasmic reticulum. Okay, so that's what happens in the process of making glucose. So as you can see, there's a lot of energy that's required. Notice that we require energy of ATP right here, going from ATP from 3-phosphoglycerate back up to 1,3-bisphosphoglycerate, reversing the glycolysis reaction. So to go from pyruvate up to glucose requires 6 triphosphates, the equivalent of 6 ATPs. And it also requires 2 NADHs right here. Okay? Now, if we go through glycolysis, the net products are 2 NADHs and 2 triphosphates. So you can imagine that if we went through glycolysis and then turned around and made glucose, every time we did that, we would lose 4 triphosphates. Okay? 6 required to make. You only get 2 out. 2 NADHs produced and 2 NADHs used in the synthesis. So the net would be we would lose 4 triphosphates if we did both of these at the same time. Now, cells don't do that. And they don't do it for a very important reason. They're not in the business of losing energy. Cells are very efficient. And so cells have what we call a reciprocal regulation system in place to ensure that as much as possible, these two processes are not occurring in the same cell at the same time. Notice I said that for the most part, the body is set up so that these two processes are not occurring in the same cell at the same time, because that will mean that cell is just going to burn energy. Now, we'll talk next term about, by the way, if these two processes are going at the same time, that is an anabolic and a catabolic process for the same molecules, that's called a futile cycle, F-U-T-I-L-E. A futile cycle occurs when exactly what I just described to you happens. Anabolic and catabolic processes are happening, and all the cell is doing is spinning its wheels, burning energy. What do you suppose would be a product of that burning? Heat. Heat is, in fact, a byproduct of a futile cycle. And our body, interestingly enough, we'll talk about it next term, uses futile cycles in some cases to actually generate heat. So they're not absolutely not there. There are cases where we actually use them to generate heat. For the most part, especially in this case, we don't want to be generating heat because we're just going to be wasting energy as appropriate. Okay, questions about that? Okay, well, let's see. Let's talk then a little bit about regulation. Okay, so regulation is important for these two processes because we want to have one on when the other one is off. That occurs by a process, as I described, called reciprocal regulation. And reciprocal regulation means that basically the same things that turn one on turn the other off. Now, there are some things that turn one on or the other on and not both. So not everything up here that you see on the screen is a reciprocal regulator, but there are a few things that are reciprocal regulators. The most important one is this compound right here, fructose 2,6-bisphosphate. We talked about it last week, and that fructose 2,6-bisphosphate, you may recall, activates PEPCK. Fructose 2,6-bisphosphate in very low concentrations also inhibits fructose 1,6-bisphosphatase. So F26BP inhibits fructose 1,6-bisphosphatase and activates PEPCK. Now, one of the things that you're going to see in the regulation I'm going to be talking about is it's very easy to get confused with all the names. Remember that whenever you see the letters ASE on the end, we're referring to an enzyme. And if it doesn't have the ASE on the end, we're not referring to an enzyme. Okay? PEPCK, of course, has an ASE on the end, phosphofructokinase, right? Okay. Now, it's important to keep that in mind as we're talking about this. So, fructose 2,6-bisphosphate turns on PEPCK, it turns off fructose 1,6-bisphosphatase. AMP turns on PEPCK, it turns off fructose 1,6-bisphosphatase. Okay? Citrate turns off PEPCK, it turns on fructose 1,6-bisphosphatase. Oh my God, how much of this do I have to know? Well, since you've got a note card.
Amen.
Okay, get the big picture. Don't get hung up in the details here, all right? The most important regulator is fructose 2,6-bisphosphate, okay? That is a very, very important thing. AMP makes sense. AMP indicates what in the cell? Low energy. Low energy, do I want to have glycolysis going or gluconeogenesis going? Glycolysis, I want to make energy, right? So I want to turn off gluconeogenesis and I want to turn on glycolysis, okay? All right, we get down here to this, and notice we're not even talking about hexokinase and glucose 6-phosphatase. We won't be discussing them with respect to reciprocal regulation, so don't worry about them. Down here, we have some things we've already talked about. Why was fructose 1,6-bisphosphate important? What phenomenon was it involved in? It was involved in pushing and pulling, but there was a better name. Feed-forward activation, anybody remember that? Right? Feed-forward activation occurs when a molecule early in the pathway activates an enzyme later in the pathway. And so fructose 1,6-bisphosphate activates pyruvate kinase. I think that's a very important one right there, okay? Because feed-forward activation is a very unusual and important phenomenon. ATP, no surprise, going to turn off pyruvate kinase. We don't want to be making more pyruvate when we've got plenty of energy. Okay? Alanine turns it off. That might seem a little odd, but actually alanine is produced from pyruvate. If pyruvate levels are high, alanine levels are high, and when pyruvate levels are high, we want to do something with it. We want to go back if we can. So we're going to turn off pyruvate kinase. Okay, so that gives you an idea about the reciprocal regulation that's involved in gluconeogenesis and glycolysis. Yes?
Why doesn't ATP turn on any of the stuff on the right?
Why doesn't ATP turn on any of the stuff on the right? Over here? The enzymes haven't evolved to that point, okay? So instead of that, we see inhibition. We see inhibition with the lower energy molecules, but no activation with the higher ones. Yep. But it's a good question. I mean, you would think that ATP would actually activate those. Enzymes don't have a binding site. Yes, sir.
Is F26BP acting competitively?
Is F26BP acting competitively? No. Allosteric regulation does not involve competitive inhibition at all. Right. So it actually, whenever you see allosteric regulation, it's much more similar to non-competitive activation or inhibition because it's working at a site separate from the active site.
Right. I was just thinking because it's very similar.
Yep. Good question, though. Good question. Okay. Let's see. Now, there's another phenomenon we have to consider with respect to regulation. And this phenomenon is, well, how in the world does the cell get fructose 2,6-bisphosphate? Kevin's already told us that it's not a product of glycolysis. Where does it come from? Well, it comes from a very interesting and unusual enzyme. I think I will convince you of that. Okay? Well, this enzyme that makes fructose 2,6-bisphosphate is an enzyme that has built into it two completely different catalytic activities. Two completely different catalytic activities, only one of which is active at any given time. Now, the activity that's responsible for making fructose 2,6-bisphosphate is called PFK2. Very simple. Phosphofructokinase 2. It's called PFK2. So PFK makes fructose 1,6-bisphosphate. PFK2 makes fructose 2,6-bisphosphate. That's simple. That's cool. Well, what's the other activity? The other activity in this enzyme breaks down, excuse me, it breaks down fructose 2,6-bisphosphate. This is why we only want to have one activity active at any given time. Now, the enzyme, the activity that breaks it down has two different names. I tend to call it fructose 2,6-bisphosphatase, but your book calls it, I I think it's FBPase 2, if I recall correctly. Let me make sure I say that right. Okay? FBPase 2. Okay. Now, this scheme tells us how the cell controls which part of that enzyme is active. So the enzyme has two different activities, PFK2 versus FBPase2. We see it starting here on the left. PFK2 is active, and FBPase2, that's a red bar going through it, is inactive. Okay? This occurs when the enzyme has no phosphate on it. So if we start out with this enzyme with no phosphate on it, PFK2 will be active, which means that this enzyme will be making fructose 2,6-bisphosphate. If the cell has its protein kinase A activated, how does that happen, first of all? One way is epinephrine, right? That kinase cascade. If protein kinase A becomes activated, it will put a phosphate onto this enzyme, as you can see here, and now PFK2 gets the red bar through it, and fructose bisphosphatase 2 is the active one, which means at this point, the enzyme is going to be breaking down F26BP. On the left, we make F26BP. On the right, we break down F26BP. When the enzyme has phosphate on it, it is breaking it down. When the enzyme has no phosphate on it, it is synthesizing it. That is fructose 2,6-bisphosphate. All right, now, well, that's important to understand. When the cell has F26BP, is it going through glycolysis or gluconeogenesis? I'll repeat that. When the cell has F26BP, are we favoring glycolysis or gluconeogenesis? Glycolysis, right? Because PFK becomes activated and fructose 1,6-bisphosphatase becomes inactivated. There's a lot of A's, there's a lot of 1,6's, there's a lot of 2,6's, and the more I say them, the more confusing to you they are going to become. So my advice number one is write these down. Learn these by writing these down. You will find it will help you a lot, because the more you hear the numbers and the enzymes, the more confused you will get. So for that reason, I'm not going to say much more about them. The important thing here is that we have putting a phosphate on makes FBPase2 active. Taking the phosphate off makes PFK2 active. Well, the last thing up here I haven't said anything about is, well, how do we take the phosphate off of here? And the way we take the phosphate off is by this enzyme right here, phosphoprotein phosphatase. Again, a phosphatase takes a phosphate off of something, and this enzyme will do it. Well, how do we determine when protein kinase A is active and when phosphoprotein phosphatase is active? It turns out epinephrine activates this enzyme. Epinephrine inhibits this enzyme. On the other hand, insulin inhibits protein kinase A, as we shall see, and activates phosphoprotein phosphatase. Reciprocal regulation carries through all the way down to this kinase phosphatase system. Now repeat that. Epinephrine stimulates the activation of protein kinase A. It stimulates the inhibition of phosphoprotein phosphatase. Insulin stimulates the inhibition of protein kinase A and the activation of phosphoprotein phosphatase. Now, you guys are looking a little tired, so I'll give you a little thing to think about. Okay? Here's the thing to think about. There's a couple of ways that we can make protein kinase A inactive. Right? How do we make protein kinase A inactive? Anybody? What does protein kinase A need to be active? Cyclic AMP. Okay? So What insulin does is it inhibits the process that leads to the activation of adenylate cyclase. Okay? It leads to the inhibition of the activation of adenylate cyclase. So the cell is no longer making cyclic AMP. Everybody with me? Well, what about all that cyclic AMP that's floating around in the cell? What does the cell do about that? It had better do something about it because there's going to be a problem, right? Even if you stop producing cyclic AMP, the cell is continuing to have this stuff floating around. Well, it turns out that cells have a built-in mechanism for breaking down cyclic AMP. That built-in mechanism is catalyzed by an enzyme known as a phosphodiesterase. So phosphodiesterase is floating around in your cell. Its normal function, it does a variety of things, but one of the things it does is it breaks down cyclic AMP. So if the cell has stopped the production of cyclic AMP, over time, the phosphodiesterase is going to be breaking down what's there, and so cyclic AMP is going to disappear. So we see with this that cells have a very good way of turning off that epinephrine signal. Okay? Now, the very cool aspect of this is that there's a compound that inhibits phosphodiesterase that many of you have sitting around in your cups. It's known as caffeine. Caffeine inhibits phosphodiesterase. Now that's pretty significant because what the cyclic AMP stimulation of protein kinase A does is it's stimulating, in this case, what? Production of glucose. In your liver is producing glucose. If you take caffeine and you keep protein kinase A active for a longer period of time, you're going to be making more glucose. One of the buzzes you get from a cup of coffee is the fact that your blood glucose levels are rising. If you put sugar in your coffee, probably even more. If you're trying Four Loko, don't even get me started. Okay? The buzzes are very real from caffeine. Now, the other buzz that happens with caffeine is that in addition to the stimulation of the production of glucose that happens, as you see here, glycogen breakdown is also stimulated. When glycogen breaks down, it produces glucose 1-phosphate, which your liver can convert into glucose and dump into the bloodstream. So there's two things that caffeine is doing with respect to blood glucose that's causing you to get a buzz when you drink caffeine. Okay. Questions about that? Yeah. The second way is that caffeine, because it's inhibiting the phosphodiesterase, is allowing cyclic AMP levels to be high, just as before. So two things happen with the cyclic AMP levels being high. Protein kinase A remains active, which means that production of glucose is increased, as well as the breakdown of glycogen is increased. The breakdown of glycogen also leads to increasing blood glucose levels. Okay. Well, it's been a while since we've had a song, so I think we should have a song, especially in view of the fact that we haven't had a song in a while. So this is one of my favorite songs. It's a kind of a long one, and I almost always get out of breath when I sing it, so I'm going to really ask for your help today with this one. It's to the tune of the supercalifragilisticexpialidocious. Oh, yeah. And it's called Gluconeogenesis. Please join me. When cells have lots of ATP and NADH2, they strive to store this energy as sugar, yes they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates. exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATP per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD, and G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory, glucose, glucose factory. The aldolase reaction puts together pieces so a fructose molecule is made with 2 phosphates in tow, and each one of these gets cleaved off by a fructose phosphatase unless F26BP is acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose jubilee, glucose, glucose jubilee. From F6P to G6P, that is the final phase. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard, because Kevin Ahern lets us use the filled out index card. Gluconeogenesis is not so very hard. That wasn't bad for me. It's a good thing I got over this cold, or I would not have done that. So, okay. So, the last thing I want to say before I move on to starting talking about glycogen metabolism is a very interesting process that occurs in our bodies, and it's one that students, I find, learn very quickly and readily. It's called the Cori cycle. It's very, very interesting cycle. So let me show you what happens with the Cori cycle. Okay? We've talked about gluconeogenesis. We've talked about glycolysis. Let's imagine that I am out for my morning run, which I was this morning. And yes, it was very slippery out there this morning. Okay? And my muscle cells are working hard. They're doing their thing, right? So my muscle cells are basically needing energy. Well, if I'm running really hard, which for me is a light jog, okay, so I'm really huffing and puffing as I'm doing my light jog, what happens is my muscle cells are needing oxygen faster than my blood supply can deliver it. We've talked about that before. That leads to fermentation. And fermentation, you recall, converts pyruvate to lactate. Why? There's no oxygen, and the cell needs to do what? Why is the cell making lactate? The cell is trying to make NAD. Okay? So it's making NAD so it can keep glycolysis going. Well, as I said in class, lactate, which is the product of that reaction, is a metabolic dead end. The cell is making all this lactate, and it's of zero use to it. In fact, if it sits around for very long, it starts lowering the pH in the cell, and it can kill the cell. So the liver cell says, well, the hell with this. I don't want this stuff, so it dumps the lactate out into the bloodstream. Here's my muscle cell. It's gone from glucose to pyruvate, not enough oxygen, so it starts making lactate. I don't want this lactate, so it dumps it out of the muscle cell into the bloodstream. In the bloodstream, what happens is the lactate moves and goes to the liver. Now the liver, as I've said before, is the major organ that's involved in regulating blood glucose. The liver is not out in the peripheral system. The liver is very close to the heart, it's very close to the lungs, and it gets the blood first. Guess where is there going to be plenty of oxygen in the liver? Absolutely. The liver is going to have a very abundant supply of oxygen. which means it's going to have a very abundant supply of NAD, which means it's not going to be going through fermentation. Okay? It's in fact going to be going and making glucose. It converts lactate back to pyruvate. And then pyruvate goes through gluconeogenesis to make glucose. The liver says, okay, I don't need this glucose. It dumps it into the bloodstream, and guess who picks it up? The muscle cells. So we see a cycle going on. And you might say, but Kevin, you said there should be no futile cycle. Remember I said within a given cell. This is occurring in the body. Muscle is doing one thing. Liver is doing another thing. This shows us very clearly how organs of our body are working in unison to provide us what we need. The liver, you're going to be, I think, by the time you finish BB451, convinced it is an amazing organ. It regulates glucose levels. Okay? It regulates fatty acid levels. It regulates fat levels. It regulates cholesterol levels. And it detoxifies half the crap that you dump into your body. It's the other half you got to worry about. Okay? It's a pretty remarkable organ. This cycle is called the Cori cycle, and it's a very important cycle for us to keep running when we start running out of energy. Okay, questions about that? No? Okay. All right, well, plodding on, and we do need to keep plodding on because we've got some territory to cover. We're going to start with the last topic of the term, which is glycogen metabolism. Now, glycogen is, as if you remember from the carbohydrates before, is a polymer of glucose. It contains only glucose. The glucose units are joined together, mostly in alpha-1,4 bonds, but about every 10 residues or so, and by the way, a residue is just one of these sugars, about every 10 residues or so, there's a branch to a 1,6 bond, as you can see here. That branch is the only 1,6 bond. So this is going from alpha-1,4 all the way across here. We have an alpha-1,6 branch, but then all the way across here, this guy is going as alpha-1,4 again. Branching is very important, okay, because what branching does is the more branched it is, the more ends of these residues that we have. Okay? Just like a tree, the more it branches, the more ends of the twigs that we're going to have, the more leaves that we're going to have. The more branches we have in a carbohydrate like glycogen, the more ends we have sticking off of this thing. Well, why is that important? So let's think back to what I said about plants. Plants have amylose, which is unbranched glucose. They also have amylopectin, which is moderately branched, about 1 every 50 residues or so, not nearly as much as glycogen is. And I told you at the time when I described that this branching was very important for the needs of animals. Okay? The needs are as follows. When we break glycogen down, we break it down starting at the ends. The more ends we have, the more glucose that can be produced rapidly. The fewer ends that we have, the less glucose can be produced rapidly. The difference between animals and plants, animals have to run away. Plants can't run away. So plants don't have the big changes in needs of glucose that plants have. Okay? A very, very important consideration. So the branching that we see in glycogen is a very, very valuable thing for us to have. Well, let's talk about how that breakdown of glycogen occurs. Okay? Here's a glycogen molecule. This glycogen molecule is shown only with the alpha-1,4s because the enzyme that I'm getting ready to describe to you does not, in fact, work on alpha-1,6s. It only works on alpha-1,4s. In fact, this enzyme only works on alpha-1,4s within about 5 sugars of a branch. So if it gets closer than about 5, it won't touch it. Well, assuming we've got more than 5 away from a branch, what does the enzyme do? Well, first of all, this enzyme's name is Glycogen Phosphorylase. Glycogen phosphorylase. We haven't talked about a phosphorylase before. We talked about a phosphatase, but a phosphorylase is doing something different. Don't confuse a phosphorylase with a phosphatase. A phosphatase takes a phosphate off. A phosphorylase uses a phosphate to break a bond. It uses a phosphate to break a bond. You can see that right here. Here's an alpha-1,4 bond between two glucoses in glycogen. Glycogen phosphorylase is catalyzing the addition of a phosphate to position 1 of this glucose to make glucose 1-phosphate. You see in the process it broke that bond. This glycogen end now has 1 fewer glucoses on it. From an energy perspective, this makes sense and is important. Why, from an energy perspective, is this important? What if I just added water and did the same thing? I could accomplish the same thing. Why does the cell use phosphate instead of water? Can it be taken off? Well, the answer is it could be, but that's not the important thing. Yes, sir?
Isn't glucose-1-phosphate and glucose-6-phosphate prevented from leaving the cell?
So you're right, but that's not the reason. But let me just say another thing about this. Glucose-1-phosphate is easily converted into glucose-6-phosphate. Does that help with the energy consideration? Okay? It's an intermediate of glycolysis. And when the cell made glucose-6-phosphate, how did it do it? What's that? With hexokinase. What was the consideration in making glucose-6-phosphate? It needed ATP. Did we need an ATP to put this phosphate on? Okay, so we did not need ATP to put this phosphate on, so we've just saved an ATP. That's good. How in the world was it that we were able to do this? Why don't cells just put phosphates on wherever they feel like it? Why is it able to do it here? It doesn't do it in other places. What does this tell you? Again, it relates to energy. Negative delta G, well, yeah, it's about, I think delta G 0 prime is around 0. It takes ATP to build glycogen. What does that tell you? Okay, think about this, folks. Yeah, Monica? There's energy stored in the bond. Okay, that's why it takes ATP to build glycogen, is because it takes energy to make that bond. We're now harvesting the energy of that bond and using it to put the phosphate on there. That's pretty cool. Okay, so from two perspectives, this reaction is important energetically. Very important energetically. Okay. Well, glycogen phosphorylase, as I said, is a very important enzyme for clipping the ends, but it's very finicky. It will not clip closer than about 4 to 5 residues from a branch. So this shows a schematic diagram of a glycogen molecule with a branch, and we can see the red guys are how far the glycogen phosphorylase will clip in before it gets to the point where the enzyme throws its hands up in the air and says, I won't go any further. Okay? To go further, the cells have to do a little dance that's kind of odd, and the dance is catalyzed by an enzyme with a mouthful of a name that we will call branching enzyme. I'm sorry, debranching enzyme. Debranching enzyme. Debranching enzyme catalyzes two interesting things. First, it catalyzes the movement of these 3 guys right here down to the end of the glycogen chain. So we see the blues going on to the end. That's the first reaction that it catalyzes. And the second reaction that it catalyzes is odd. It uses water to clip the 1,6 bond right there. My pointer is going out, okay? The 1,6 bond right there between the green and the yellow is clipped using water, not phosphate. What does that tell you? Anybody? Quickly. There's not as much energy in a 1,6 bond as there is in a 1,4 bond. Therefore, we don't have enough energy to put a phosphate on there. We have to use water to clip it off. This is the only place in glycogen metabolism where free glucose is released. Everything else releases glucose 1-phosphate. Okay, I think I'm out of time. Let us pick that up next time, and I'll see you guys then. Hey, Andy.
Well, it didn't work this morning.
Really?
Yep.
The computer?
Don't do anything.
Yeah, I'm not. Yeah, I won't.
Good, that's perfect.
Yeah. It's worked fine for me. Of course, I have my own jump drive.
None of my audios or video. I did too.
Huh. Anyway, I haven't had any problems. So,
You know why that didn't, it's going to come up, right?
are you sure it's on your drive?
Oh, no, I'm just saying, didn't it just come up Windows or Apple?
I think it resets before it does that.
Oh, no. Oh, there you go.
Yeah, there you go. Good. You should be fine.
Hi, what's on the test? I'll ask it for you. Ask it for you.
I was just wondering, when's the last day to submit the regrade? Is it Wednesday? Because you have the 3 full school days. Okay.
I have a notepad question. Can I glue graph paper to it to make it neater?
No, it has to be all by hand.
Okay.
Good question.
I took my test early.
Uh-huh.
And I was wondering if I could pick it up.
Oh, that's right. Yeah, we screwed up where your test was. I've got. In fact, I talked to my wife. You gave it to her, right?
I gave it to the TA.
To the TA. Okay.
Yeah.
The TA gave it to my wife, and when you sent me an email, like, oh my God, my wife has it. I apologize. I will get. I haven't graded it yet. I will get it. Do me a favor. Send me an email saying, Kevin, give me the damn exam,
Okay.
and I will get it for you by tomorrow.
Okay,
Okay.
thank you.
Sorry about that.
For the problem,
Okay.
for the problem that you said the TA should have given us one time, do you want a formal written thing for that as well?